This lecture is entitled Psychoneuroimmunology, the Basics. My name is Mark Hutchinson. I'm a research fellow funded by the Australian Research Council at the University of Adelaide. I'm the leader of the neuroimmunopharmacology lab within the School of Medical Sciences at University of Adelaide. We're a group of researchers that are funded by various funding bodies such as the Australian Research Council, the National Institutes of Health in the US, as well as the US Department of Defense. Our research interests lie in three main disciplines of pharmacology, immunology, and neuroscience. The reason I'm telling you this is that the following lectures will really be based out of our statistical, molecular, preclinical, and clinical experiences in the field of psychoneuroimmunology and neuroimmunopharmacology. And so we will be broaching topics at both the molecular and clinical ends. So what to expect? So in this lecture, hopefully we'll cover the brain to immune and immune to brain communication, just some basic concepts around this and how, and hopefully you'll see towards the end of the third lecture how this is quite important for understanding both how opioids work as well as just generally how chronic pain is communicated. We're going to talk about the specific cell types that are involved in this, especially around those immunocompetent cells within the central nervous system, microglia and astrocytes primarily, also touching on some of the roles of peripheral immune cells. We'll discuss a little bit about oligodendrocytes and clearly also, which may be surprising to some of you, the immunology of neurons and the capacity that neurons have to both signal in an immune fashion as well as receive immune signals. What are these signals that are communicating between these various immunocompetent cells? Well, they're your classic immune signaling molecules, cytokines and chemokines, as well as various other small molecules and larger patterns that we'll talk about. Finally, hopefully we'll start to capture why this all occurs. Just in the basic concepts, really we still don't fully understand the evolutionary reasons why these processes are in play. We perhaps have inklings into the reasons, but we don't fully appreciate yet the total complexity of why evolutionarily we have these systems. But at a molecular standpoint, we do have some of the key milestones in the pathways understood, and we'll talk about them today. In the second lecture, we'll focus on how immune to brain and brain to immune communication goes wrong, specifically relating to chronic pain, the involvement of the various immunocompetent cells within the central nervous system, how the cytokines and chemokines play a role in this, as well as some of the other newer molecules such as the danger-associated molecular patterns. The reason why this occurs, the big question, we don't know. And this is still a hot topic of research. But what these discoveries have enabled us to do is to start to develop new types of treatments specifically targeting the immune dysfunction that is associated with chronic pain. And we'll talk about some of the basic concepts around those new treatments. And finally, in the third lecture, we'll touch on what happens with immune to brain communication with opioid analgesics. And how the glial component of this system goes wrong, and we'll talk about some of the mechanisms about how this goes wrong, and what the opioid-induced glial pro-inflammation within the central nervous system does to change what's happening within the spinal cord. Finally, in lecture 3, what we'll do is we'll touch on the impact that opioid changes in immune function within the central nervous system has on the action of the opioids, hence the opioid pharmacodynamics, how opioids modulate the various functions of immune cells within the spinal cord and higher centers, the signals, the immune signals that are generated by opioid action at these immunocompetent cells, and touch on perhaps some of the reasons why this occurs. We still don't fully understand this. But excitingly, these discoveries that have been made at the preclinical and clinical level are paving the way to new interventions to augment and perhaps improve the analgesic efficacy and safety of opioids. So today we're going to be talking about psychoneuroimmunology and touching on some of the neuroimmunopharmacology. What we're dealing with is the unification of multiple systems. Really what we've previously thought about in terms of neuroscience is that behavior is mediated by the wiring of the body, the neuronal system, modulations in this system purely restricted to the brain. What psychoneuroimmunology suggests is that the immunology of the system of the organism out in the periphery is not just partitioned out into the periphery. Immune changes, immune activation with to change behavior by modifying neuronal systems within the brain. And interestingly, of course, now with the advent of understanding the immunology of the brain, this barrier of the blood-brain barrier is simply just not there, and there's the capacity for the immunology of the brain to drastically modify the behavioral outcomes of neuronal firing. Of course, where do drugs fit into this? Well, a lot of CNS targeted agents were previously thought to just act within the brain, as they have their primary receptors perhaps targeted within the brain. However, we understand now that yes, this is a key direct action within the brain, but there may also be some indirect pathways where drugs are able to modify the function of the immune system, either central immune system or peripheral immune system, and this indirectly is able to change behavior, either beneficially or detrimentally. And so there may be situations where a pathology actually resides within the central immune compartment that is presented phenotypically as a behavioral problem. And drugs have been designed to target the putative neuronal mechanisms of this behavioral problem. And yet the pathology persists in the immune compartment. And so this understanding, breaking down these walls between these three compartments, has enabled us to examine far greater extent this almost holistic or systems physiology approach to various pathologies. But to start with, we're going to talk about psychoneuroimmunology. And this is this fascinating interaction between the brain and the immune system, and how the immunology of the organism is able to change behavior by interacting with the wiring of the body. So if we talk to start with about the wiring of the body, where is there evidence to suggest that the immune system actually is able to be modulated by a brain to immune communication? So if we follow the neuronal innervation of organs down to the organs, what we can see is that even primary immune organs such as the spleen are innervated by sympathetic neurons, suggesting that there is the capacity of the brain to modify immune responses just by this straight neuronal innervation. Clearly, that's not the only way that the body's immune system can be modified, and of course there's the endocrine route of activation or modification of the immune system. And many immune cells express receptors that are compatible with the endocrine pathways of the body. And this is, of course, another way with which the brain can modify immune responses. So I hope some of you are thinking, oh, what's he going on about? Surely this is just common sense. What one has to remember, though, is that up until the probably mid-80s to early 90s, the neuroscience domain were operating in complete isolation from immunology, and immunology did not really take into account brain action. And no models were being developed in order to understand how this particular communication may actually occur. We know circumstantially that there is evidence to suggest that stress perhaps may lead to increased risk of infection, for example. But there wasn't any hard data available to demonstrate this link. Now, this history of brain to immune communication is fraught with many tears and broken careers. The Western world wasn't aware that, in fact, the Russian scientists in the 1920s, several groups, were working on Pavlovian conditioning of immune responses. And this work really had remained hidden from the Western medicine up until the mid-90s, when it was finally rediscovered. But in terms of Western medicine, George Solomon, a psychiatrist, coined the term psychoimmunology when he proposed that there was this very important interaction between brain processes and the immune system, and specifically he was referring to particular psychiatric disorders and the associated immune dysfunction that was related to that. But these were very controversial ideas that were put forward, and in the context of the environment that George was talking about in the 1960s. This actually led to him being completely shunned by the scientific community. His ideas were suggested to be far too controversial, far too outlandish to be believed. And this almost forced him to completely give up his research career and just maintained his clinical practice. But it wasn't until the 1980s, when the AIDS epidemics really took hold, that his ideas began to be taken seriously and actually tested. And if it wasn't for George Solomon's original work, most of these concepts would have been delayed much, much further. And he really bore the front of the great opposition against this domain, which is now quite a well-established research discipline. What has made this research discipline well-established is the hard data, if I can call it that, demonstrating a profound link between the brain and the immune system. The first hard data that was presented was that published by Robert Ader and Nicholas Cohen. Robert Ader, a psychologist, and Nicholas Cohen, an immunologist. And what they did was they looked at taste-conditioned immune suppression. What they used was a pairing of saccharin, a sweet solution that has no caloric benefit, co-administered with cyclosporin, clearly an immunosuppressant agent. And they did this on repeated conditionings. Various times of number of conditionings were provided, either no conditioning, 1, 2 conditionings, and then looked at, on the final challenge, the impact that just exposure to the saccharin had on an immune response. In this particular case, it was an antibody response. And what they showed was that just the pairing of the immunosuppressant agent cyclosporin and the saccharin resulted in a profound ability of just exposure to saccharin to produce an inhibitory response in the immune system. So I hope you can realize that we're talking about saccharin, which is just activating taste sensors, hence activating some form of a long-term memory consolidation, probably hippocampal perhaps, resulting in a change in the immune function out in the periphery. And so these were the first data to demonstrate that yes, the brain could actually fundamentally modify in a specific context related situation the immune system. Now, subsequent to this, a lot of studies have been done. Some of them you may be aware of. For example, we know now that environmental and psychological stresses are both profoundly modify the immune response. So we know, for example, that environmental stress is associated with increased infection rates, and there's some great studies by Glia in 1985 showing that medical students prior to examinations and then during examinations had very different immune reactivity with the stress of the exams resulting in decreased immune function. However, it's not that stress completely decreases the function of the immune system. It's in fact a biphasic response. So when stress first kicks in, there's actually an acute elevation in the capacity of the immune response to fight, and as such likely to be part of the fight or flight response, and this leads to an acute potentiation of the capacity of the immune response. If, however, this stressor lasts for a longer period of time, this results in an inhibition of the immune function, leading to decreased capacity to respond to infection, and hence increased rates of infection, perhaps, and also clearly linked to decreased clearance of cancer. So what, as I've just mentioned, this brain to immune communication is extremely important. It's quite very important for placebo effect, clearly, stress and disease, such as the cancer story I was just talking about, and is now being linked with the depression comorbidities as being a very important contributor to increased rates of various inflammatory or infection associated with depression. So that is brain to immune. And I guess if we look at the wiring of the body, we can understand that makes sense. But what about immune to brain? Can the immune system talk back to the brain? So the classic example of this is, how do we know we're sick? If we have the flu, we know we've got the flu, we feel lethargic, we're often more sensitive to light, we don't want to eat as much. And so we know our immune system has to be able to be communicating with our brain, otherwise we simply wouldn't know that we were sick. And these, of course, have beneficial, evolutionarily beneficial effects, such that we're conserving energy, we perhaps are resting our wounded limb, for example, or infected limb, and so on. How does this occur? Well, we know that this occurs owing to some great research done in the past 20 years, that cytokines, such as interleukin-1, are able to communicate back to the brain. A classic example of this, that the brain is able to be modified by the immune system, is some work in the late 90s by Serge Rivest. And what Rivest's group did was they looked at the brain activation of I-kappa-B, which is associated with the NF-kappa-B pathway, following systemic lipopolysaccharide or endotoxin exposure. What you can see here is the basal activation status of I-kappa-B in vehicle-treated animals, so there's really not a lot of activation going on. However, after the systemic administration of LPS, you see that there is a profound activation in multiple brain centers at 1 hour and 3 hours following LPS, but starts to get resolved at 6 hours. And this is quite widespread throughout the entire brain. What Rivest's group then looked at was the particular regions within the brain, the CVO regions of the brain that are leaky, and showed that IV administration of LPS or IP administration of LPS both resulted in the activation of this I-kappa-B pathway in these leaky regions of the brain. They hypothesized that LPS administration or exposure should be driving a pro-inflammatory cytokine response, and so they postulated that if they then just gave these pro-inflammatory cytokines, they should also see this activation of this pathway within these brain regions. And sure enough, what they saw was that tumor necrosis factor alpha and interleukin-1 beta both were able to produce this profound activation of the I-kappa-B alpha pathway within the CVO regions of the brain. Subsequent to this work, several new ways of brain communication, immune to brain communication, have been postulated and demonstrated. The type that we were just talking about there with Serge Rivest is the humoral pathway, where activation of peripheral immune cells leads to the release of pro-inflammatory mediators, which go up to the circumventricular organs of the brain, which results in the translation of a pro-inflammatory cytokine signal into a prostaglandin and nitric oxide signal. And this has been elaborated on further into various other pro-inflammatory signals originating from this site. These pro-inflammatory signals can then lead to changes in behavior. The second type of signal that has been demonstrated to occur is that of vagal nerve signaling, such that the pro-inflammatory cytokines are able to be detected by the vagal nerve, and this vagal nerve activation up to the NTS leads to the translation into a central pro-inflammatory response originating from the NTS, and again producing behavioural changes. And finally, probably the newest member of this communication pathway is the cellular route, where pro-inflammatory signals are translated up via the CVOs as potentially other sites as well, leading to a conversion of this pro-inflammatory signal into a pro-chemotactic signal, such as MCP1 or monocyte chemotactic protein 1, which then calls some of these specific subsets of these immune cells into the brain to help with and perhaps control or perhaps direct and coordinate this immune response within the brain. And this is quite a fascinating line of research with some very interesting implications. So how on earth does this immune response, as I talk, or immune signaling, actually change the brain? Well, clearly what we know is that it is fundamentally changing the brain. We know it's occurring in wide disparate regions, so we should be able to be seeing modifications in neuronal function. We know, as I mentioned, that pro-inflammatory cytokines such as interleukin-1 are key molecules, but how does this actually work? And I think what we need to touch on first is this difference between central immune signaling versus neuroinflammation. Now, I know a lot of you will have already been quite au fait with the term neuroinflammation, but perhaps less so with central immune signaling. And I think it's an important distinction. Neuroinflammation really is associated with some gross pathology, such as a trauma or injury, such as the pathologies we've got with multiple sclerosis, Alzheimer's, stroke, etc. This is where we have significant, profound changes in the immune system within the brain and can probably be imaged with standard imaging techniques. I've got chronic pain here because there is, unfortunately, I believe, the use of neuroinflammation associated with chronic pain. However, I do not believe you were able to actually image with standard techniques chronic pain-induced neuroinflammation. In contrast, what I'm talking about with central immune signaling is a different part of the spectrum of neuroinflammation, down towards the more discrete, subtle volume control side of the system, where these changes are occurring on a daily basis without any major perturbation of the system and potential to be reset without significant damage occurring to the brain. So in order to understand this, we need to understand a little bit more about what this basal central immune signaling and homeostasis is within the brain. I hope you can all remember seeing a picture like this, beautiful images of a neuronal cell body with the axons projecting out and some very nice electrical activity represented here with the red highlight. However, I hope you also realize that this image is completely wrong. We don't have large amounts of space within our brain, and that space is made up by the immune-like cells of the brain, those being the microglia, astrocytes, and oligodendrocytes, as well as the blood-brain barrier and small amounts of peripheral immune cells. And all of these are capable of profound immune signaling. What are these glia? Just to give you a quick reminder, they are the non-neuronal cells of the central nervous system. There are an awful lot of them. 70 to 90% of the cells in your brain are in fact these glia. In fact, this is where we get the misconception that we are only using 10% of our brains. It was the fact that the media misreported the journal article where they were doing a survey of the neuronal to glial ratio, and they saw that 10% of the brain was neurons, and clearly we only use our neurons. We don't use any of our glia to do anything. And this is where they reported that we only use 10% of our brains. But we now understand that that's completely not true. Glia are absolutely pivotal to the maintenance of CNS homeostasis. Neurons are unable to survive without glia because of their trophic support. And we now know that these astrocytes, pictured here, microglia and oligodendrocytes, are absolutely fundamental to normal brain function, and unfortunately they do go wrong and contribute to pathology as well. How do all these cells work together? Well, we know that astrocytes are pivotal in maintaining synaptic function. They're involved in maintaining the blood-brain barrier. Microglia act as the sentinels, responding to damage very rapidly, and oligodendrocytes ensheath the axons to assist with efficiency of signal transmission. Now, just to go over those in a little more detail, microglia, they account for about 15% of the cells. They're very reactive, highly mobile. They are phagocytic, so they can taste the microenvironment of the brain, but they actually do have a distinct origin from the peripheral macrophages of the peripheral immune system. They have the full array of immune detection systems, and they can release immune signals once they become activated. And as an example of this, what we have is a green fluorescence protein transgenic mouse, which has been bred specifically to have a thin skull, so these authors in the science manuscript could do two-photon electron microscopy through the skull. And what you see here is the activity of microglia in real time, shooting out processes, sampling the microenvironment. And what we have over here is a zoomed in portion of this showing this sampling of the microenvironment within the brain. And it's estimated between about 20 to 25 minutes. Every 20 to 25 minutes, your brain is actually, all the nooks and crannies of your brain is sampled by microglia, actively surveying the homeostatic potential of the brain. Microglia are also very responsive to damage. In this particular example, a laser beam is shot into this region of the brain, again using this animal model. And what you can see is the rapid migration of microglia to the site of injury from quite disparate regions in a very rapid fashion. Astrocytes, they're the most abundant cell type in the CNS. They perform a variety of structural roles, the blood-brain barrier. They maintain the tripartite, tetrapartite synapse. We'll talk about that in a moment. They have huge number of connections with up to 30,000 other cells, often both oligodendrocytes, microglia, and other astrocytes. This is very important for the sensitization of other lateral sensitization and cross-sensitization of other pathways. We'll talk about that in a moment. They provide trophic support to neurons. They're also capable of immune signaling, but they're a little slower off the mark than microglia. And really, it's probably only once microglia have told the astrocytes that they can signal in a profound immune fashion that they actually do. But they do have this capacity, because they have this large network of connections, they're able to signal via calcium waves. And this is an area of fascinating study at the moment, demonstrating sensitization of parallel pathways via calcium waves through astrocytes. Some of you may remember when you were taught your neuroscience, the presynaptic and postsynaptic images, talking about the vesicles leaving the presynaptic terminal and acting on receptors on the postsynaptic terminal, and how it was a lovely system, but there was nothing wrapped around it. Well, what we now understand is that astrocytes are key participants in about 70 to 80% of synapses within the brain. They ensheath these synapses and actively control what is actually going on in the synapse. So they are sampling it, they are taking from it and adding to it. Under certain situations, this tripartite synapse with the pre, post and astrocytic end foot is complemented by the addition of microglia, sampling and adding to the synapse. And this has been coined by Joyce DeLeo's group as the tetrapartite synapse. Now, what we know about these astrocytes is that they have these wide array of other connections, up to 30,000 others. And fascinatingly, if a particular synapse is undergoing a large amount of activity, that particular astrocyte that is ensheathing that synapse will become activated. And that activation can result in calcium wave propagation, such that that astrocyte ensheathing another synapse can sensitize a parallel, related or unrelated synapse neuronal network, resulting in lateral signaling and sensitization, such that we have now the reinforcing, perhaps, of a particular signal and the translation of activity in one pathway to another via an astrocyte process. So this process of the tripartite system modulating the neuronal activity is a very important concept when we come to actually understanding how immune activation in the periphery translates to fundamental changes in neuronal activity through modulation of the tripartite cooperativity. How does immune activity of a microglia modify the function of an astrocyte? Well, one of the first things that happens after microglia become pro-inflammatory and release pro-inflammatory cytokines and chemokines is that the astrocyte loses its ability to maintain the glutamate transporters on its cell surface, and especially around the astrocyte infeed of encapsulating synapses. And what we see is that these two profound pumps, the GLT1 and GLAST, which account for up to as much as 1% of total CNS protein, these become down-regulated during times of pro-inflammatory response in microglia. And what this results in is an acute upregulation in glutamate levels. Now, clearly, if we have acute upregulation of glutamate, this can lead to a neuroexcitatory environment and even a neurotoxic endpoint. So if you're interested in this concept of blood to immune, microglial to astrocyte, astrocyte to neuron, this review by Miller in 2009 is a fantastic place to start. What's important to realize is that this inflammatory response could originate from the peripheral blood, or in fact it could originate from a microglial perturbation of the system. Oligodendrocytes, they are the cells that ensheath the neurons to maintain the efficiency of action potentials. Each oligodendrocyte is capable of ensheathing up to 50 axons, again providing a capacity for cross and lateral sensitization of pathways. Unfortunately, they are prone to this neuroinflammation, less problematic with central immune signaling, and this is clearly a problem in multiple sclerosis, for example. Oligodendrocytes have been shown to have some capacity of signaling, clearly receiving immune signals, but some in actually propagating immune signals. But this is, again, much slower than microglia and less advanced than astrocytes. Now, surprisingly, you may see here neurons with their capacity for central immune signaling. As it turns out, neurons are just not neurons. Neurons require cytokines and chemokines to be able to maintain basal function. Cytokines and chemokines are both required and fundamentally employed by neurons to maintain states of both LTP and LTD. And without them, the neuronal system is locked in either states of constant LTP or constant LTD. There's quite a profound dysregulation of the system in cytokine such as IL-1 knockout animals. What we also know is that this immune signaling within the brain can be quite selective. So, for example, the CX3CL1 or fractalkine signal, which is expressed within the CNS only by neurons, binds only to CX3CR1, which is the fractalkine receptor, and within the central nervous system, this is only expressed by microglia. So we have a very nice example of a neuron to glial communication signal, which is one of the only ones that's in existence that we know of. Of course, the barrier that protects the brain from the peripheral circulation, the blood-brain barrier, is critical in translating or not translating a peripheral immune response into the brain. Tight junctions are maintained to stop large molecules getting across, such as cytokines. But there are parts of the brain, such as the CVOs, where this blood-brain barrier is intentionally leaky. Of course, the blood-brain barrier is made up of these endothelial cells, which, as you know, have immune capacity. So there is now evidence to suggest that a pro-inflammatory signal on the blood side of the circulation may actually translate into a pro-inflammatory signal via endothelial cell translation into the brain side. And finally, peripheral immune cells. Now, this is an area of evolving work. Previously, we would have thought that there wasn't migration of T cells or B cells or macrophages perhaps into the normal healthy brain. And most of the time this is probably the case. We don't have large numbers of these cells migrating into the brain. However, under states of pathology, and perhaps not so much pathology, and perhaps under states of what I'll term central immune signaling, there may be the requirement of and requesting of the blood-brain barrier and the immunocompetent cells within the brain for the presence of peripheral immune cells to support and help the response. So an example of this would be the selective recruitment of particular T cell subsets into the central nervous system. So I hope you can see that there is this clear distinction between central immune signaling and neuroinflammation. And the neuroinflammation really is this profound end, damaging end of a spectrum where we would expect to see swelling and increased edema, as well as a large amount of damage occurring. However, central immune signaling is much more of the everyday phenomena. We would expect central immune signaling to be involved in homeostasis and perhaps stretching into not quite right, but let's not ring the alarm bells just yet. So central immune signaling is very important for trophic support, crucial for maintaining basal neuronal functions, and for perhaps maintaining the recovery from certain small types of CNS injuries. So what are the factors that we've been talking about that these cells release? Well, you'll know cytokines and chemokines, reactive oxygen species that are profoundly used by the central immune system, and various others. Newer ones that we're beginning to understand are these endogenous danger signals, or otherwise known as danger-associated molecular patterns, and we'll talk some more about those in the second lecture. The cytokines, clearly we have our pro-inflammatory and anti-inflammatory cytokines, those being the pro-inflammatory ones like interleukin-1 beta, interleukin-6, tumor necrosis factor alpha, and many others. And of course, this is in a yin and yang system. We have anti-inflammatory cytokines such as IL-10. These cytokines have the capacity to act either directly or indirectly in the brain, as we discussed. And they alter multiple different types of activities of neuronal functions. So we have the modulation of neuroendocrine function, we have modulation of synaptic plasticity, we have changes in transmitter turnover, and a lot more. So just to summarize this first lecture, what we've covered is the basics of immune to brain and brain to immune communication. We've talked about the specific cell types of the brain, some of the basic concepts around how an immune response in the brain may actually change the neurochemistry of the brain, and some of these signaling molecules we've just touched on briefly. Thank you.